All right. Disclosures. I'm on the scientific advisory board for MediBeacon that's developed this technology. I'll briefly restate what the shortcomings of an estimated GFR using an equation are. First of all, there's a considerable lack of precision that results in misclassification of patients. It doesn't, the MDRD and CKD-EPI equations do not apply to all populations. They're only valid in the populations used to derive the equations. So you need truly multiple equations if you're going to do, for example, multinational clinical trials. And of course, and most important for this meeting, they cannot be used in non-steady state conditions, something that many of our hospitalists and surgeons fail to remember. Again, non-GFR determinants of creatinine are multiple that include differences by age and gender, race, body habitus, the effects of illness, and you've heard already the effects of even a short stay in the intensive care unit in terms of loss of weight and muscle mass and decreased production of creatinine. So these all make creatinine in the setting of a non-steady state a very inaccurate and effectual measure of kidney function. So the ideal GFR agent, as you know, is something that does not undergo metabolism. There's no protein binding. It's eliminated totally by the kidney and through no other pathway, filtered by the glomerulus, excreted only as a result of that filtration. There's no resorption or tubule secretion. And ideally, we would like to have a way to use this ideal GFR substance in the non-steady state in critically ill patients. Bruce has mentioned some of the methodologies that are out there. They basically fall into three categories: urinary clearance technology, plasma clearance technology, and nuclear. They all have their shortcomings. Urinary clearances generally involve the need to instrument the bladder so as to avoid incomplete emptying. Minimum collection period for doing sequential creatinine clearances, for example, is probably an hour, and some have suggested 2 hours. And you need to have some measure of the serum marker, and so if you're in the steady state, maybe one measurement is sufficient. If you're in the non-steady state, you need to bracket the urine collections with 2 blood draws, so you can measure twice. Plasma clearances, there are a lot of inaccuracies, as Bruce mentioned, with a single sample. And usually they require a long duration of multiple sampling to derive that plasma disappearance curve. And nuclear requires, obviously, a detector, and there's radiation exposure, not amenable to doing this repeatedly, and certainly not appropriate in the context of critically ill patients. So this is the solution that MediBeacon has come up with, and I will talk about each of these in turn. They have a patented fluorescent tracer agent that I'll describe in a moment. They have a sensor that can be put on the skin, and in the preliminary trials, this sensor has been put on the thigh, the forehead, the sternum, the arm, with consistent readings from all of those sites, and they have a real-time monitor. The compound currently is called MB102, and these are the properties of this compound. It's very hydrophilic, it's biocompatible, it has no metabolism, as you'll see in a moment, excellent solubility, very good stability, so shelf life of this compound will be very long, and it doesn't, it's not sensitive to any photo instability. And importantly, it has a large Stokes shift. What is that? Stokes shift means that you excite this compound at a wavelength of 434, and it returns an emission at 557, and that difference between the wavelength to excite the compound versus the wavelength that it emits when excited allows for a very good signal-to-noise ratio. The compound has a molecular weight of 372, and I just put, for comparison, some of the molecular weights of our other compounds that are frequently used to determine glomerular filtration rate. So this lies somewhere between creatinine and iohexol. Iohexol is the gold standard for plasma disappearance curves currently. And of course, this is much lower than either dextrans or inulin, which is not a single molecular weight, and certainly much lower than cystatin C. What do we know about this compound? Well, in the preliminary trials in humans, these are 12-hour urine collections. 99% of this compound comes out in the urine, similar to iohexol, the gold standard. So we believe that this is eliminated from the body totally by renal excretion. I'll show you half-life in some subsequent slides. This is a rat study just to show you what happens after a bolus injection. The dark line represents the serum fluorescence, transdermal fluorescence, whereas the dotted line here is the accumulation in the bladder. And you see that the compound distributes throughout the body and very quickly starts to accumulate in the bladder. Again, just reflecting the fact that this substance is renally excreted and has no other pathway for elimination. You inject the compound and there is a single peak. So this is the peak determined 30 minutes after injection of the compound. So there are no metabolites that are generated. This is a totally inert substance. Now, what's the decay curve look like for this MB102? This is time along the x-axis, and this is the concentration determined from plasma sampling. Each one of these circles is a blood sample. This is in humans now, and you can see that there's this elimination fits two-compartment model where there's a rapid decrease in the plasma level as the compound distributes in extracellular space, and then a slower slope that reflects the elimination through the kidney. In this example, after a single injection of a few ml of this material, the window for continuous measurement here is 10 hours. It doesn't take 10 hours to get a GFR measurement, but one can continue to monitor GFR through the exponential decay here for a window of that length. Now, to show that this is truly a GFR marker, we need to compare it to the gold standard, and so that's what's done here. These are simultaneous plasma sampling of the marker over here on the y-axis and simultaneously injected iohexol. And you can see that the identity line, the regression line between the two samples matches very closely the line of identity, suggesting that the decay curve for this MB102 is identical to the decay curve of iohexol, the gold standard for a GFR marker. Now, go to the transdermal methodology. I will show you this cartoon. Basically involves a LED light that shines the excitation wavelength through the skin. A sensor, in this case on the sternum, picks up that wavelength, puts it through some filters that allows it to process just the 557 nanometer wavelength, which is then converted from analog to digital and put into a computer. And I show you now the fluorescence PK versus the plasma PK. So to start with, here's the plasma levels. Each of these circles represents a plasma sample. These, again, is all in human patient with, this represents their skin color. So this has been done in patients with varying skin colors to prove that that will not confound or limit the use of this. So here's the two compartment decay curve, nice and linear. Here's the fluorescence now, superimposed on the decay curve. So again, these are the samples, blood sampling, and each of these little dots is the fluorescence, showing that it's fitting the same curve. And so when you put those together, there is an identical decay curve for the fluorescence as there is for the plasma sample. So in essence, the fluorescence can be used instead of plasma sampling to determine this exponential decay. And as I said earlier, we've done this in a variety of skin types, all showing these excellent fits between plasma sampling and the fluorescent decay curve. And I would point out that this compound MB102 has a half-life of approximately 2 to 2.5 hours in individuals with normal kidney function, which would suggest that the window during which continuous GFRs can be recorded after a single injection is going to be somewhere between 4 and 6 hours in people who have normal kidney function, and considerably longer in patients who have impaired kidney function. This is just a correlation between, again, the plasma and the transdermal fluorescence of this substance as done over a number of hours in a variety of patients, showing this very tight correlation between the transdermal fluorescence and the plasma measurements. So these decay curves are identical, and that means that using transdermal fluorescence to generate the decay curve, and then knowledge of the dose that's administered and the volume of distribution, which can be extrapolated from the decay curve or using anthropomorphic criteria, allows a calculation of the GFR. And right now, we're thinking that that's GFR would be calculated every 15 minutes. And importantly, this is repeatable. So when you get down to the end of the decay curve, if you wanted to repeat this and continue the monitoring, you just give another dose. This is what the curves look like in different levels of kidney function. And there's a current trial, both here in Europe and in the US, that will be comparing MB-102 fluorescence and plasma disappearance in patients with a variety of levels of impaired kidney function. And this is what the monitor in its current state looks like. It will communicate with your multi-channel monitors in the ICU. It'll display the GFR. It'll display the history of GFR. Right now, this is set for roughly every 15 minutes. It will tell you the time here at the bottom until the next injection might be necessary. So that's the secundi, and a little bit of dessert. Why would we want to do this? Well, in the non-steady state, daily monitoring of measured GFR would be very useful to guide antibiotic and other drug therapy. We have no idea, for example, how to dose antibiotics in patients who are on CRRT, or when to start calcineurin inhibitors post-transplant. Early determination of a decreased GFR would guide our use of how to use the KDIGO bundle. It could be a novel determinant of the adequacy of dialysis dose. How about if instead of looking for KT over V or ml per kilo per hour as a metric, we put down equivalent GFR? Might determine when to stop dialysis in an ICU patient, guide deresuscitation in the ICU. When does GFR start to go down when you're aggressively removing fluid with diuretics, as was discussed this morning? Will we have to go back to the rifle criteria for AKI if we have measured GFR? Only rifle had a GFR criteria for AKI stages. What about in the steady state? Well, we'll have more accurate assessment of GFR and staging for chronic kidney disease. That may improve chemotherapy dosing. It may turn toward a different endpoint for clinical trials, something that's going to be discussed subsequently. There's been a lot of conversation over the last day or two about measuring renal reserve. I think this methodology lends itself very well to that opportunity. We could determine whether renal reserve then is a risk factor for progression, whether patients who recover from acute kidney injury have lost renal reserve, maybe a better way to accept patients for organ donation. We could look at the response to pharmacologic therapy and perhaps predict whether patients are going to get a favorable response to use of ACE inhibitors or angiotensin receptor blockers. And we might be able to determine the trajectory of residual renal function and its loss in incident dialysis patients. So in summary, MB-102 is cleared solely by the kidney. The plasma clearance matches exactly the plasma clearance of iohexol, which is the current gold standard for measured GFR. And the plasma clearance of MB-102 matches exactly the fluorescent clearance of MB-102. And thus, the plasma fluorescence can be used as an adequate marker of GFR. The GFR can be obtained every 10 to 15 minutes once distribution in the extracellular volume is achieved, and that takes about 45 to 60 minutes. And it can be monitored continuously for 4 to 10 hours, depending on the baseline level of kidney function. This compound's adaptable to continuous measurement of GFR, analogous to glucose clamping, and it may now enable us to actually have a true renal vital sign. And this potential development by the company of a sublingual method for administration that would facilitate outpatient usage. Thank you.